Okay, let's see now how some real data look like. So in this example, what I mean real data here, I mean some data from sequence data using the Illumina technology, and this data will be I think exome data, so it will not be from the whole genome, but does not really matter actually for us now. It will be only from the coding regions, and they are from a plant called glycine max or otherwise soybean. So this is the plant. So from this plant we took some genetic material and we applied all the methodologies, I mean all the technologies that I described in previous videos, and then we extracted DNA or RNA, doesn't really matter, and then we sequenced that using Illumina. Okay, now to get, so when a scientist applies all the sequencing technologies in his lab, then usually they upload the data in some servers. There are three servers around the world, the three main servers around the world that they upload data. One is the NCBI. So this is in the US, and the specific server that hosts the data is called SRA. If I remember well, Sequence Read Archive or Short Read Archive, I think it's Sequence Read Archive. Then there is another one in Europe called ENA. And in Europe, this ENA stands for European Nucleotide Archive. And there is another one in Japan, and I think it's called let's see how it's called. This in Japan is called DDBJ. So when a scientist produces some data, he may choose where to upload the data. For example, it's very reasonable here in Europe to upload that in ENA. And when it's uploaded in ENA, it will take a code. The run that we put in Illumina, it will take a code, which usually starts with ERR, if this is in the ENA repository. So this read then, this result, this data, in other words, will be transferred automatically to both NCBI and DDBJ, and will keep this code, which for example it might be ERR 15695. This is just an ID for the sequencing results. That the data that the scientist uploaded will take automatically. This is a unique identifier for specifically these results, and if the scientists upload this data at ENA, it will take a code starting with ERR. Okay, on the other hand, if somebody will upload data to NCBI, it will take a code starting with SRR. And then this code, for example this code here, will be transferred to both ENA. So ENA will have then SRR something and DDBJ. I say that in order to mention that when you download data from NCBI, for example, which is the American server, sometimes you will notice that data results start with SRR, other times with ERR, and other times with DRR. And this means that firstly this data will were uploaded at NCBI here, or to ENA here, or to DDBJ here. Okay. So regarding the glycine max data that we will use, if I go to NCBI, so I will use this ID here, SRR 391539, and this is some sequencing RNA-seq data. We will talk about RNA-seq a lot during the class, but for the moment it's not really important because the data format will be exactly the same. And here they did some experiment where they compared the response to chronic ozone of 5 agriculturally important legume species. One of the species is glycine max, soybean, and here are the results. So the header here informs us that this is an Illumina experiment using Illumina machines. It's HiSeq 2000, which, if I remember well, it produces single or paired end reads of 100 base pairs on each side. So if you use paired end, it will be 100 plus 100, 200 in total. But as far as I see here, we have single end, see just a forward direction. If there is also a paired end, then usually you see an arrow pointing backwards here. So these are the data from the reverse direction. It informs us here that it's glycine max, and has different codes here. So there is this code, for example, that starts with SRX. There is this code that starts with SRP. There is this code that starts with SAMN, and finally there is this code that is interesting for us that starts with SRR. So what are all these codes here? So if we go here to some help page, so to how I found it, I googled SRA, what is SRX, and SRR, and then the first result is this one here. Okay, and informs us that there is a code starting with SRP, which is a study, which means actually it's a study, and this is a set of experiments that have a specific goal. In this study we might have several experiments, so this is the set of experiments here, several experiments, each one will have a different SRX code, and then each experiment might have several samples, and this sample starts with SRS code. An experiment targets one or more samples, here they are. Results are expressed in terms of individual samples or bundles of samples, because it's possible that we have pooled several samples together, as defined by the experiment. Then we have the SRR, which actually means a result. Results are called runs. Runs comprise the data gathered for a sample or sample bundle and refer to a defining experiment. Okay, so the results are stored in some file which its ID starts with SRR. Okay, so we have the SRR file here. And now I will show you how it looks like. So when I downloaded the SRR file, I think this is that's the ID, just to write it here. It's 391539. 391539. I downloaded this from NCBI, and this in NCBI is stored in a specific format called SRA, if I remember well. So it has this SRA suffix. This is not the data that we usually analyze we don't use in analysis, in our analysis. Instead, we convert this data directly from NCBI to the so-called fastq format. So this fastq format will be the raw format for us, and this is what I will show you today, because this is actually our raw format. Now, if we will use NCBI, for example, then we will convert the SRA format to fastq, but this I will demonstrate that in another video. It's not actually a proper video for the first lecture. And then again we will go to fastq and we will start from fastq our analysis. Okay, let's now see how fastq looks like. Alright, so I opened a fastq file, specifically this SRR file 391539 that we saw before, and here it's the first read actually that you see. So let's start from the beginning. And a fastq file contains the information for several reads, for all reads that we got. So let's do it like that. I will just copy paste this information in the notepad, in the notebook, just to be able to comment it immediately there. So let's go back here and let's zoom in. Okay, so just copy paste some values there. So this is a fastq file and contains information for all reads that we got from sequencing. So in fact we expect to see DNA data here. Instead we see some other lines as well. Now I will explain what they are. The read information, so the sequencing, comprises one entry. So one read is actually one entry here. And this entry has different information, carries different pieces of information. Actually 4 pieces of information. The first one is this line here. That's a header line. And starts always with an @ So for example here it says that this is a read with ID SRA 391539.1. It has this, whatever code it is, this from the machine, and its length is 100. We don't really need it. So this is the first line of the first entry. So this is entry number 1. And then on the second line of the first entry, this again entry number 1, but the second line, there is the actual sequence. So this tells us that this read starts with a C, and then it's a T, 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 G, A, C, A, G, A, A, and so on. The third line, the important thing is that it starts with a plus. And then there are some other numbers, some other codes, which usually it's the same like the first line, but it's nothing important. So what we need here is a plus. And then the fourth line assigns a quality to each read of a base. So for example, it informs us here that this C was read with a quality H. And I will explain that in a while what it means. In contrast, this A here was read with a quality encoded by the sign equal. Or this G here is read with a quality encoded with a sign greater than. Okay, so these quality symbols actually, quality symbols corresponds to numbers. And in fact they corresponds to, if I remember well, to 40 numbers. Okay, let's see what they are actually. I go back to Google here. I just type fastq Wikipedia. I go here in fastq format. And somewhere here at the bottom of the page there is this table. Okay, let's see if we can zoom in. Okay, maybe a bit more. So the strange symbols that you could see before are these symbols here. And in Illumina that we use, this dark purple, whatever it is, it's this L here, we have these 40 qualities. So for example, the exclamation mark is quality 0. The quotes, the double quotes, is quality 1. The I is quality 40, which probably is the best one. So this means that if a base pair, let's go here, this say here that has a quality equal is somewhere here, it's actually read with a quality corresponds to the value 88 as 28 or so on. Now we usually trust qualities that are above 30, which means that we don't really trust so much this A here. Now the next question probably will be, okay, why to use these strange symbols to represent qualities and not use numbers from 0 to 40 to represent the qualities? The reason is the following. If we go here to the raw data, we see that the representation of qualities as symbols allow us to have a one-to-one association of the base that we have called and the quality itself. So we see directly here that this C corresponds to this H, and this C here corresponds to the quality C here, or this G corresponds to the quality hashtag, or this G to the question mark. On the other hand, if I would have numbers from 1 to 40 or from 0 to 40, then I could not have this association, because a quality 11 will occupy two characters here, 1 and 1, and then a letter here would occupy just one character. So I would not be able to align properly the sequence and the reads and the qualities. If I would have to do that for a computer, probably I would have to say something like that. If I have the A, C, A, G, A, for example, and I would like to assign numbers as qualities, I would say, for example, 11, 12, 35, 28, 3, 4, 5, 17. Now you see that actually to represent the qualities I need several characters here and comma values, which means that such a result would be would occupy much greater space in a computer. And for this reason, a more compact representation has been chosen that will not have any comma values or any gaps here or whatever. It will just have a one-to-one alignment between base pairs and qualities. So for example, here it could be H equal H A B, and then we are fine. Something else that we can notice here is that different technologies or even different versions of Illumina here or different other companies have different quality score representations for the reading. The software that we will use for downstream analysis actually can understand what the machine, what the encoding is, and they can judge if they want to use a low quality read or just exclude it from further analysis. For example, a software might choose, or we may choose, by applying the proper filters in a software to exclude reads that are of low quality. So for example, we will just neglect the fact that we have read an A here because the quality of A is low. And we'll say, okay, I see an A here, but I'm not sure what it is because the quality is not very high, so maybe I will put an N instead of an A, meaning any nucleotide can be here, or even, that we will see that later, with a process called trimming, I remove all the initial reads up to a certain length because usually they are of low quality. We will see that later on in a different video. And then so with these processes we try to keep only the part of a read that comprises higher quality score values. Okay, so the first entry, as I said before, comprises 4 elements. So here comprises 4 lines. And then we have the second entry, which is the second read up to here. And then the third entry up to here, and then the fourth entry up to here, and so on. And usually we have several millions of reads in such a file. That's all for this video. I hope everything was clear. Thank you very much.